Hello, welcome back, all of you. In this session, we'll be discussing the second chapter of first year, namely the biological classification. In the first chapter, we have seen that classification is one of the prime necessities in studying living things. We cannot study biology, we cannot study about living things without classification. And right from the time of Aristotle, people tried to classify organisms. But at that time, our knowledge on living things were very poor and they were not accurate or scientific, and they classified organisms based upon their use. And all classifications at that time and later on were based upon visible external morphology. That is, external characters that can be seen from outside. So they need not be true or correct always. But later on, as we attempted to study living things with more accuracy, more scientifically, there developed different types of classifications. The study of classification is called taxonomy, that is, norms regarding taxon formation. Norms means rules and regulation in which we should classify group organisms into different taxa. And the father of modern taxonomy is none other than Carolus Linnaeus. His classification was that there will be 7 taxa, 7 obligate categories, of which the largest taxon is the kingdom. He made 2 kingdoms, though the terms Plante and Animalia was present right from the time of Aristotle. Linnaeus made them as his largest taxa. So, this classification is called 2 kingdom classification. What are all Plantae? It included plants. But there is a problem. This Plantae included unicellular algae, bacteria, then ferns, bryophytes, modern advanced plants, etc. Similarly, Animalia included all animals. They were again made up of, or they were having unicellular organization or multicellular organization. All of them were coming under the same category. So, this differentiation is mainly based upon 2 important things, namely whether the cells are having a cell wall or whether they are having chloroplast and performing autotrophy, photosynthesis. That was the main criteria in grouping them into Plantae. Those which are having no such cell wall and photosynthetic ability were grouped under Animalia. Irrespective of the nature of organization of the body. Sometimes body will be unicellular, sometimes it may be multicellular. That was the 2 kingdom classification, which is having a number of such discrepancies. Unicellular organisms like protozoa, in which amoeba, paramecium, etc., were coming along with mammals, which are highly developed, advanced, multicellular. Similarly, Chlamydomonas, chlorella, which are all unicellular algae, are all also included under multicellular dicot plants. So there was such a discrepancy, but it was very easy to remember and a simple type of classification. But taxonomist after Linnaeus modified this two kingdom classification into three kingdom. It was Haeckel who separated unicellular organisms from multicellular organisms and produced a third kingdom, namely the Protista. The Protistants included all unicellular organisms, whether they are all autotrophic or heterotrophic. Irrespective of that, if their body is made up of a single cell unicellular organism, then they were all put under the Protista. So there will be three kingdoms in addition to Plantae and Animalia, Protista. And again, Copeland identified that all unicellular organisms are not alike because some of them are having a well-defined nucleus, membrane-bound structures like mitochondria, whereas others are having no such structures, no well-defined nucleus, no membrane-bound structures. So he separated all such organisms which are having no true nuclear membrane, no membrane-bound structures in the cells as monerans, and those which are having only true nucleus with membrane-bound structures alone are put under Protista. So a fourth kingdom was thus produced by Copeland. What is that? It is Monera. What are monerans? Which are all containing only unicellular prokaryotes. That is a word that was coined later on by Fort. What is a prokaryote? Pro means primitive. Karyos means nucleus. Having a very primitive type of nucleus, having no membrane of its own, no nuclear membrane, no membrane-bound structures, and unicellular. That is the feature of the monerans. Whereas Protistants are containing unicellular organization but with a well-defined nucleus and membrane-bound structures. And in 1969, we made one more addition that was done by R.H. Whittaker, an American taxonomist, who separated fungi. Till this time, fungi was only a group coming under Plantae. He studied and noted that fungi are different from Plantae in a number of respects. And so, it is not justifiable to include these fungi in Plantae. So he separated them and elevated to the rank of a kingdom. Fungi was till this time only a class coming under Plantae. Now he elevated them to the rank of a kingdom. Thus made the fifth kingdom. So this classification that Whittaker proposed is called five kingdom classifications. What are the five criteria that Whittaker took for his classification? He took five criteria. Of these five criteria, number one, the most important one is what is the nature of the nucleus. nuclear organization. Whether it is prokaryote or eukaryote, all prokaryotic organisms are coming under monera, and all eukaryotes were grouped or separated from this one. That group is again divided based upon the number of cells in the body. That is called thallus organization. How their body is made, whether it is single cell or multi-celled in nature. All unicelled organisms are coming under the protista, and multicellular organisms were separated, and these multicellular organisms were again classified mainly based upon the type of nutrition. If they are all autotrophic, they are belonging to plantae. If they are all saprotrophic, they were all belonging to fungi. And if they are all heterotrophic in nature, they are all belonging to Animalia. And this separation was done also taking into account the reproduction. There is a considerable difference between plantae and fungi. That is why he separated fungi. Mainly, plantae are containing organisms whose body is a sporophyte, diploid. Whereas in fungi, the thallus, the body is haploid, gametophyte. Okay. In plantae, the meiosis takes place during gamete formation. So it is gametic meiosis. Meiosis occurs during gamete formation. And these gametes are taking part in fertilization. Whereas in fungi, meiosis takes place in zygote. It is zygotic meiosis. Okay. As a result of that, again, haploid spores are produced, which will be growing to a fungal thallus, which is haploid. Okay. So, there is major difference between plantae and Animalia. Sorry, between fungi and plantae. And lastly, all modern classifications are taking phylogenetic relationship, evolutionary relationship as an important criteria, because we know that all these organisms came to this world from a very, very common ancestor once upon a time through millions of years of evolution. So if organisms are related to each other, similar to each other, they should also have the same evolutionary history. That evolutionary history is phylogeny. See, in grouping organisms, we are taking into consideration whether they are similar or not. If they are similar, they are put under the same category. And if they are similar, they should have similar evolutionary background. They have undergone the same way of evolution. They are phylogenetically connected. So, phylogeny is a number one criterion in almost all modern classifications. So, he took the nuclear organization, the thallus organization, nutrition, reproduction, and phylogeny as the 5 criteria in his classification. Okay. Now, this classification is having some advantages. What is the advantage? Here, unlike the two kingdom classification, there is separation of unicellular organisms and multicellular organisms. Unicellular organisms are again divided into prokaryotes and eukaryotes. All prokaryotic unicellular organisms are coming under Monera. Okay. Then we are having roughly I can draw the relation between the five kingdoms Whittaker has made in this diagram. What is this diagram depicting? See, monerans are the first formed organisms which are all simple. And from this Monera, the Protistants have evolved. And from Protista, Plantae evolved, and later on through another line Animalia evolved, and fungi also came into existence later on, different from Plantae. See, Plantae and fungi had a common ancestry. Later on they bifurcated, they went evolving into different ways. So that simple diagram is sufficient to explain the evolutionary relationship also. Here, Protistants are having unicellular. They are all unicellular but eukaryotic. Monerans are also unicellular but prokaryotic. So there is the first difference, whether they are all prokaryotic or eukaryotic. And among the eukaryotic, we are dividing them into unicellular and multicellular. That is the second level. This is unicellular and these are all multicellular. And third one, nutrition. Plantae are all photosynthetic, that is autotrophic. Fungi are all saprotrophic, and Animalia are heterotrophic. They are all heterotrophic. So we are having the five kingdom classification represented diagrammatically in a graphical form, in a symbol form, which is also showing the evolutionary relationship. Clear? This will be having the problem of two kingdom classification solved. What was the problem with the two kingdom classification? There was no separation of prokaryotes and eukaryotes. There is no separation of unicellular, multicellular organisms, but now that is solved. Now, Plantae are all containing only multicellular, eukaryotic organisms, whereas Chlorella, Chlamydomonas, which are unicellular, are separated. Okay. Among Animalia, you are having Paramecium and Amoeba, which are all unicellular, put under the kingdom Animalia in two kingdom classification. Now they are separated and they are all brought under the Protista. So we are having plant-like and animal-like. Then you may have a doubt. How is it correct to include plant-like and animal-like organisms? The answer is very simple. What is our criteria? Organization of the body. All of them are having unicellular. They are unicellular, eukaryote. That is the criteria by which we are all classifying. So, based upon that, we are grouping plant-like and animal-like organisms under the same category. They differ in their nutrition, but that is not our main criteria by which we are all classifying. This classification is based upon the thallus organization. Clear? So, this five kingdom classification is widely accepted, and we are now studying this five kingdom classification. Are you ready? Okay. The first kingdom is kingdom Monera. What is the criteria of organism to be included under Monera? They should be all unicellular. They are all unicellular, made up of only one cell, unicellular, and prokaryotic. Simple structure. Though they are all very simple, remember, they are the first to be evolved in this world. So, the ancient, most ancient kingdom is Monera, because Monera contains bacteria, which are all unicellular prokaryotes. So, bacteria are the sole members, only members coming under this category. They are all present everywhere, and they are noted for their maximum metabolic diversity. They show maximum metabolic diversity. They will be autotrophs. Among autotrophs, you are having photoautotrophs, just like plants. They are having some pigments, and based upon the pigments, they will perform photosynthesis, though it is slightly different from that of the photosynthesis in plant A. They perform photosynthesis, some of them. Some of them are chemoautotrophs, preparing chemicals. They perform chemical reactions and they get energy out of that reaction. They are called chemoautotrophs. Some of them are parasitic. They are living on both plant and animal bodies as parasites. Some of them are saprotrophs. They are doing a very valuable job of recycling of minerals. They cause the decay of dead bodies, converting them back into simple inorganic minerals. So they are all acting as decomposers. And many of them are living as heterotrophs on others. Okay, so most majority of them are the heterotrophs. Bacteria can be grouped into four based upon their shape. If they are spherical or globular, coccus, cocci plural. Bacillus, singular. If they are all rod shaped, elongated, bacilli, plural. Spirillum, if they are all coiled like a spring, spirillum. And if they are coma-like, it is called vibrio. It is called vibrio. So this classification is mainly based upon the appearance, the shape of the body. Now, they are having very simple structural organization, but they are showing maximum metabolic diversity. Most extensive metabolic diversity is shown by them. They are classified into archaebacteria and eubacteria. Archaea means ancient. So, they are the first formed bacteria which are still remaining without much change in their body nature or in their physiology. That's why they are called living fossils. They can be called as living fossils, and they are all occupying very amazing situations or living in extreme conditions. Like they may be living in marshy areas, they are called methanogens, or in extreme salty areas called halophiles, or living in hot springs called thermoxylophiles. So they are capable of living even in such conditions. Remember, our world was like that once upon a time when they evolved. Still, they are all remaining without much change, living the same condition. How they are all capable of living in such extreme hazardous conditions? Because there's a speciality in their membrane. Their membrane is different from that of our plasma membrane. Their membrane is made up of unusually long, highly branched fatty acid connected to the glycerol by either bond. So we can simply say that fatty acid connected to glycerol by either bond. In our case, fatty acids are connected to the glycerol by ester bond. Here it is either bond. So the cell wall structure is different. Their cell membrane is made up of fatty acid connected to glycerol by either bond, and they have a cell wall which is mainly made up of proteins. That is the speciality in their cell wall. So they are having a different cell wall that enables them to live in such extreme conditions. Okay. Methanogens are present in the gut of ruminant animals like cow and buffaloes and are responsible for the production of methane gas. So we are using this bacteria for producing methane gas, gobar gas out of cow dung. Okay. Now, eubacteria. Eu means true. It is true. There are true bacteria. Archaea means old, ancient. So, true bacteria are having a different cell wall. Their cell wall will be having what is called peptidoglycan. Peptidoglycans, which is also called as commonly murine. It is murine. So, they are characterized by the presence of a rigid cell wall. That cell wall is having peptidoglycans or murine in them. That is the main difference between archaebacteria and eubacteria. And a eubacteria can be of different categories. One type is cyanobacteria. Cyano means blue-green. They were all showing some resemblance to the algae. So we thought that they were all algae, and so the name blue-green algae or cyanobacteria is given to them. But we now understand that they are all different. What are the similarities? This cyanobacteria is having chlorophyll A, just similar to that of plants, and thus they are all autotrophic and photosynthetic. Though they are all unicellular, they will be forming colonies. These colonies will be very often elongated filamentous structures, which may be living in both freshwater, marine, or in terrestrial conditions. And these colonies are having a jelly-like covering over them, just like algae, which is giving a slimy touch. When you touch it, it will be slimy because of the mucus covering. So we thought that it is alga. Okay. And they will be forming blooms in polluted water bodies, just like algae are all forming blooms, they will be multiplying and producing large number of them, filling the entire surface of water bodies when it is polluted. Some of them are having a special cell in them called heterocyst. In this heterocyst, you are having a special ability for atmospheric nitrogen fixation. They can absorb nitrogen from atmosphere, and atmospheric nitrogen is converted into nitrates. Such cells are thus increasing the nitrate availability in the medium. So they can be used for increasing nitrate fertilizer ability, or nitrate fertilizer can be produced in the soil by exploiting these types of bacteria, cyanobacteria, because they will be growing in them and will be producing nitrates in the soil by absorbing atmospheric nitrogen. And they also perform a large number of inorganic chemical reactions such as nitrate conversion. So they will be utilizing nitrates, nitrites, ammonia, and they will be releasing the energy, utilizing the energy from these biochemical reactions for their own use. They are called chemoautotrophs. Okay. They are playing an important role in mineral nutrient cycle like nitrogen, phosphorus, iron and sulfur. And most of the bacteria are heterotrophs. They are not autotrophs but heterotrophs. And some of them are decomposers, and these decomposers are helping in decomposing the dead bodies, that is in recycling of nutrients. Again, they are helping in making so many things that are needed for us. Some bacteria are helpful, beneficial for mankind. Okay, some are living in roots of higher plants, providing nitrate to them. As symbiont, they are all living, both of them are benefited, both plant is benefited as well as the bacteria are benefited. Bacteria is getting some carbohydrate material from plant, and the plant is giving accommodation to the bacteria. Now, the bacteria is giving nitrates to the plant, which is very essential for the plants. So both of them are getting benefit. Others are pathogens. They will cause diseases, and we are having a large number of diseases like cholera, typhoid, tetanus, citrus, cancer, etc., which are all caused by bacteria. Now, how the bacteria is dividing? Usually, bacteria are dividing or multiplying by binary fission. When conditions are favorable, they will be reproducing by binary fission. That is the commonest method. But when conditions become unfavorable, they will produce spores, so spore formation. They also reproduce by a type of sexual reproduction, which is called conjugation. A sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to another during conjugation. In conjugation, two bacteria may be coming close together, and after coming close together, they may exchange their genetic material, namely the DNA will be exchanged between them. Such a sexual type of reproduction is rarely seen. Then another type of bacteria are mycoplasma. They are also called PPLOs, puro pneumonia like organisms. Okay. They are having no cell wall. That is the main difference. Since they are having no cell wall, there is no definite shape. Without shape, meaning they will go on changing their shape. They are pleomorphic. They can change their shape, pleomorphic, and they can survive without oxygen, and they are the smallest organisms with cellular organization. They are having no cell wall, but they are having cell membrane, the primitive type of nucleus-like structure and other structures by which they show that they are having cells. So, smallest one with cellular organization is the mycoplasma or PPLOs. Okay. All of them are parasitic either in plants or animals, and they are anaerobic. Anaerobic. Okay. So that is about the first kingdom, namely the Monera. Then we are having Protista. Protistants are, what is the criteria? They are also unicellular, but they are all eukaryotic, having a well-defined nuclear membrane, chromosome, and the cells are having membrane-bound structures. So, this Protista is a heterogeneous group, meaning this contains plant-like organisms, animal-like organisms, fungus-like organisms, and bacteria-like organisms. So, the boundary of this kingdom is not so clear. Okay, this includes plant-like, animal-like organisms. This kingdom forms a link with others, dealing with plants, animals, and fungi. We have seen in that diagram, we have discussed that it is from the Protista that fungi, animalia, plantae, etc. have evolved. So, this is a link. To other groups of organisms like plants, animals, and fungi. Okay. Now, what are the features of this one? They are having sometimes a cell wall, sometimes they may be absent, sometimes they may be autotrophic, sometimes may be heterotrophic. So, we are classifying them into 5 groups based upon mainly 3 features. What are the features? I will tell. 1. What is the cell wall? Here in Chrysophyta, the cell wall is made up of cellulose which are impregnated with silica. So, we are having silica, siliceous cell wall. Number 2, they are all photosynthetic. So, photoautotrophs. Okay. Since they are the photoautotrophs which can go on multiplying very quickly, they are the most important producers of ocean. They are all floating passively in water current. Such organisms are called planktons. The most important plankton of the sea are diatoms. These diatoms and golden algae, which are having pigment which are golden in color, that is why it is called golden algae. They are called desmids. They are the most important members coming under this one. Their siliceous cell wall is indestructible, so that diatoms have left behind large amount of cell wall deposits in their habitat that will be accumulating over billions of years to form what is called diatomaceous earth. What is the advantage of this diatomaceous earth? What is diatomaceous earth? The cell wall. You know what is diatoms? Diatoms are called so because the shell wall is made up of two pieces, just like our soap dish. Just like our soap dish. And when the organism leave this siliceous cell wall, they will be falling on the sea bottom, and after thousands and thousands of years of pressure, they will be transformed into a special type of gritty soil. Soil that is very gritty, rough in nature. That is called diatomaceous earth. So being gritty, very rough in nature, it is used for polishing, for filtering oils and other thick syrups, and their main significance in ecosystem is that they are the chief producers in the ocean. Clear? So that's about the chryso. Chryso means golden, golden algae. They are having pigments. Okay. Now coming to the second one. In the second group, we are having dinoflagellates. These dinoflagellates, why they are called dino? Dino means spinning. When they are all moving, they go on in a spring-like way. They will be having a spinning movement as they move. They go on rolling. That is why it is called dino. Dino means spinning. They are all exclusively marine. Okay. They have again cell wall. That cell wall is made up of stiff cellulose plates. These stiff cellulose plates are arranged end to end or placed close together so that they will be leaving a groove in between, a shallow groove in between. Lying in that groove, you are having two flagellae. One flagellum lying lengthwise and the other flagellum lying across, round. That's why they are having a spinning movement. When this flagellum, that is called trailing flagellum, is vibrating, the animal moves forward. When the transverse flagellum vibrates, the organism goes on spinning. So, because of this, you are having a characteristic shape, a characteristic movement. That's why they are called dinoflagellates. So, they are also having cell wall. Number two, they are also photoautotrophic. They will be having different colors for their pigments. So, depending upon the nature, they may be having green, yellow, brown, blue or red coloration. Okay. And these red dinoflagellates like goniolacs undergo rapid multiplication that the entire sea surface will become covered with them. What is their color? Red pigment. And thus they give red color to the water. So it appears as if the whole sea water is covered with red carpet. Such a condition is called red tide. They produce toxins which are highly dangerous to other organisms. The toxin is called XI-XII. XI-XII. Okay. That is strong enough to kill even other marine animals like fishes. Okay. Toxins released by such large numbers will be strong enough even to kill other animals. Now coming to the euglenoids. Euglenoids are freshwater. They are all freshwater. In stagnant waters like potholes, small ditches, pools, etc., they have a cell wall which is made up of protein called pellicle. That pellicle is flexible. It is flexible. Protein is the main component of that pellicle. Okay, so flexible pellicle that allows them to change their shape, and they will be having two flagellae, one long and one small. Now coming to the nutrition, that is the second point. One is the cell wall covering. Two, nutrition. They are also having chloroplast, but they will be also capturing some other cell debris and small organisms as their food and live like an animal. So they are photosynthetic, and at the same time they are also heterotrophs. Thus they show what is called mixotrophic nutrition. They show what is called mixotrophic nutrition, both plant-like and animal-like. So we say that they are representing a connecting link between Plantae and Animalia. They are all mixotrophs. Okay, mixotrophs. When there is sunlight, just like plants, they will be performing photosynthesis. When there is no sunlight, no problem, they will be capturing food with the help of the small tentacle, small flagella. The pigments are similar to those in the Plantae. Okay. Now coming to the next group, namely slime mold. What is slime mold? They are commonly called protistan fungi. They are all protistan fungi. Why they are called so? They are fungi-like because they are all saprotrophic. So it's called protistan fungi. Protistan fungi. They will be moving along the decaying twigs and leaves. They are all depending upon decaying materials. So where they are present, they will be growing, and there is no size and shape for their body. Their body is called plasmodium. Under suitable conditions, they will form an aggregation of structures called plasmodium, which is having no size and shape, but a large number of nuclei will be present that may grow and spread over several feet. But when conditions become unfavorable, that is when drought comes or there is no decaying materials available as their food, when conditions become unfavorable, the plasmodium produces small fruiting bodies within which they will be producing spores. These spores are having true cell wall. Slime molds are having no cell wall. That is why they are slimy to touch. That is why it is called a slime. Mold means fungus. Fungus which are slimy to touch. That is the reason why they are called slime molds. Why they are slimy to touch? Because they are having no cell wall. But their spores are having rigid cell wall, true cell wall. And because of the cell wall, they can resist adverse conditions and may live for many years even under adverse conditions. The spores are spread by air currents, only through air. And the last category are animal-like protists, which are called protozoans. We believe that it is from these protozoans that modern animals have evolved. So, primitive relatives of animals, they are having no cell wall and are all heterotrophic. So, of these 5 groups, Three are autotrophs. What are they? Chrysophyta, Dinoflagellata, Euglenoidea. One is saprotroph, that is the slime mode. And the last one is heterotroph. Their nutrition is exactly like animals. So it's called holozoic. They may be living as holozoic organisms, taking in complex food material. They may be living as predators or they may be living as parasites, okay, on the body of other organisms. We are having four major groups of protozoans based upon the type of locomotor structure. We are classifying them into flagellated protozoans, ciliated protozoans, amoeboid protozoans, and sporozoans. Amoeboid protozoans are having pseudopodia, false, temporary feet, as in amoeba. Some may be having a shell made up of silica. Some are living as parasites. In flagellated protozoans, long hair-like flagellum. They may be also free living or living as parasites. For example, the sleeping sickness causing parasite is trypanosoma. It is a flagellated protozoa. Ciliated protozoans, they are having a large number of small hair-like structures called cilia. And they have a cavity that opens to the outside the cell surface. The classical example is the paramecium. See, in paramecium, which is called slipper animalcule, having the shape of a slipper. See, it is having the shape of a slipper. On one side, you are having a groove. Through which food is taken in. The whole body is having cilia. Okay. And the coordinated movement of the cilia cause the movement. Okay. And also brings food from outside into the vestibule or to the gullet. Param is a classical example for ciliated protozoan, commonly called slipper animalcule. Slipper animalcule. What is the meaning of that word? Cule means a small. So, a small animal having the shape of a slipper. And we have sporozoans, meaning spore-like. So, it is based upon the locomotor structure that we have classified into the pseudopodia like flagellated protozoans, ciliated protozoans, etc. Sporozoans are spore-like. That is why they are called sporozoa. And they will be having an infectious spore-like stage in their life cycle. They are all parasites and they don't have a locomotor structure. The notorious parasite, Plasmodium, that causes malaria, is coming under this category. Sporozoans are having a spore-like stage in their life cycle. They are without any locomotor structure. So, that is the classification of the protozoa. So, protozoa is the fifth group coming under protista. Is that clear? The 5 groups? Now, we pass on to the kingdom fungi. Formerly, this was included along with plantae. Now, they are all separated. Because plants are having diploid sporophytic stage in their life, which is the most important stage in their life, sporophytic generation. Whereas in fungi, the soft body. The soft body is called thallus. The thallus is made up of large number of branching tube-like structures, thread-like structures called hyphae. They are all haploid. Their nutrition is saprotrophic or parasitic in nature. Many of them are parasites. So either living as saprotrophs, majority of them are saprotrophs, responsible for breaking down of complex organic materials, whereas few are parasitic. Okay, these fungi are mostly multicellular, but there is one exception. What is that? The Saccharomyces, which is commonly called as the yeast, is unicellular. Baker's yeast, Saccharomyces, is unicellular. So in them there is no thallus. Instead, they will be undergoing multiplication and will be producing a number of unicellular organisms which are remaining connected together as a colony, and thus it forms what is called pseudomycelium. So Saccharomyces is having a pseudomycelium. Only one fungus which is unicellular is Saccharomyces. Most of the fungi are heterotrophs and absorb soluble organic matter from the dead substrate. Thus, they are all saprophytic. But others are parasitic both in plants and animals. A few of them will be remaining in association with the algae as lichens, or along with the roots of higher plants like mycorrhiza. Both are typical example for symbiosis. They are living in mutual benefit with either algae or with higher plant root. So they show symbiosis. Okay. Now, these are belonging to so many groups and we are classifying them based upon three features. One, morphology of the mycelium. That is number one. Two, how spore formation and fruiting bodies are occurring. So, second one is spore formation. Three, fruiting bodies. What is a fruiting body? A body in which the zygote is undergoing meiosis resulting in spore formation is called fruiting body. It is within the fruiting body that zygote is undergoing meiosis and spores are produced. So, how spores are formed and what is the nature of the fruiting body? These are the three criteria on which they are all classified. Fungi can reproduce by vegetative mechanisms, by asexual mechanisms, or by sexual mechanisms. During sexual reproduction, there are three stages. What are they? In most of these fungi, there will be fusion of protoplasm between two motile or non-motile gametes. Fusion of cytoplasm is called plasmogamy. Gamy means fusion. Fusion of cytoplasm is called plasmogamy. Then, after that, only the two nuclei will be fusing. That's called karyogamy. Karyos means nucleus. And the third and the last one, this zygote thus formed undergoes meiosis. Remember, I told you that is the main difference between plant and fungi. In fungi, meiosis takes place after sexual fusion. It is the zygote that is undergoing meiosis. It is zygotic meiosis. So, meiosis in zygote resulting in haploid spores. These spores are growing out and thus you will be having haploid hyphae producing. Now, when a fungus reproduces sexually, two haploid hyphae of compatible mating type, that is matching type, come together and fuse. In some, the fusion of the two haploid cells immediately results in formation of a diploid one, whereas in others it may form a structure which is having two nuclei remaining together without fusion. The cytoplasm is fusing, but nuclei are not fusing immediately. Thus, you have a cell with two haploid. This is also haploid, this is also haploid, because this is formed by the fusion of two haploid gamete like structures. So, this stage is called dikaryon phase or dikaryon. So, this condition is called dikaryotic stage or dikaryon, meaning having two haploid. Di means two, karyon means nucleus, two nucleated condition. Later, the parental nuclei fuse. These two will be fusing and thus it becomes diploid. Immediately it undergoes the fungi form fruiting bodies in which reduction and division occurs, and that will be resulting in the formation of again haploid spores. So, we can simply write, we are having two haploid spores that will be resulting in a dikaryotic one. Thus, it finally becomes 2n that undergoes meiosis. And as a result of meiosis, you have haploid spores. There are haploid spores. These spores develop into a hypha, and in the hypha, you will be having male and female gametes, two types of gametes. So, life cycle is very simple. The main part of the hypha, that is the haploid generation, gametophytic generation. This is the hyphae, which is formed by the growing out of spores. Is that clear? Now, this is classified into 4 categories. What are these 4 categories? Let us study in a comparative way. We'll be making columns. Simple. It is phycomycetes. The term phyco means algae. So, they are commonly called algal fungi because they are living in wet places. They are called algal fungi. This is the only group where the hypha is a syncytium or phenostium. What is that condition? Here, the hypha is having no septa, aseptate, meaning no septa, and there will be numerous nucleus in it. So, aseptate and branched hypha, that is number 1. They reproduce asexually by producing either flagellated zoospores or non-flagellated eplanospores. Both these spores are produced within this sporangium. So, we call it as endogenously produced. That is the asexual reproduction. Asexual reproduction is by zoospores or eplanospores, which are all produced inside the sporangium. So, it's called endogenously produced. And sexual reproduction is by zygospore formation. Sexual reproduction is by zygospore formation, which is also produced within a bag or a sac-like structure called sporangium. So again it is endogenous. These spores are also produced endogenously. Our bread mold, rhizopus, mucor, and a parasitic form will be the classical examples coming under this. That parasitic form is causing a disease. What is that disease? Mustard spotting or mosaic disease on mustard. That is the albugo. Albugo is causing white dots on the leaves. That's why it is called spotted or mosaic disease in mustard. So these are some examples. Clear? Now, coming to the second one. What is the second one? Please note. The second example or second group actually is ascomycetes. That is the largest. Okay. It is commonly called asco means sac. So it's called sac fungi. Why it is called so? I will tell you in a minute. Here the hypha is not syncytium. Hypha is branch and septate. Branch and septate. Okay. They are reproducing by conidiospores. These conidia are produced on the outside. So, it is called exogenous. Conidia are asexual spores. Asexual reproduction is by conidiospore formation. And during sexual reproduction, spores are produced in a sac-like structure, which is their fruiting body. So, inside that fruiting body, what is that fruiting body? Sac-like fruiting body. Inside that, the sexual spores are produced. So, it is called the sac fungi or ascomycetes. Okay. So, you will be having the spores produced inside the fruiting body. The fruiting body is called ascocarp. Inside ascocarp, 8 spores. What is the name of the spore? Ascospore. 8 ascospores are produced. Fruiting body is the ascocarp, within which 8 ascospores are produced. Since they are all produced inside, it is endogenously. So, we are having exogenous asexual reproduction occurring, resulting in spores, whereas sexual reproduction resulting in spores which are produced endogenously. And this is the largest group. Okay, this is the largest group in which we are having Saccharomyces. Okay, which alone is having a single celled body. Other examples are Aspergillus, Claviceps. Claviceps is causing the rye of, it is a parasite on the ergot of the rye and commonly called ergot of rye. Neurospora is genetically used in researches and so is called the Drosophila of plant kingdom. Remember that this was under the plantain, so the old name is still retained. Some of them are edible, like morels, morchella, truffles, esculenta. Okay, they are all edible and are very, very tasty in nature. The third one is basidiomycetes. Why it is called basidiomycetes? Basidium means club. They are commonly called club fungi. They are commonly called club fungi. Here the septum is there. So here also you are having branched and septate type of hypha. Remember, only phycomycetes you are having syncytial condition. In all of this, it is septate. Okay. They reproduce only sexually, no asexual spores. No asexual spores. They reproduce only sexually, but no sex organs. They usually reproduce by plasmogamy. We have studied plasmogamy, where you will be having two cells, vegetative cells fusing together. Their cytoplasm will be fusing, but their nucleus will be remaining as two separate ones, dikaryon stage. That dikaryon grows out into a secondary hypha that will be developing into the fruiting body. It is that fruiting body that we call as mushrooms. See, why this group is called lichens? Lichen means mushroom. Mushrooms are the most common and most important members of this group. That's why this group is called fungi. Okay, so mushrooms are the classical members coming under basidiomycetes. That mushroom is their fruiting body. On the mushroom, you will be having a large number of fruiting bodies called basidiocarp. Fruiting body is called basidiocarp. That will be producing 4 basidiospores. These basidiospores are produced exogenously. Outside, there is fruiting body, but spores are produced on the outside, on the fruiting bodies. Only 4 basidiospores. So understand the differences? Here 8 ascospores, here only 4 basidiospores. So mushrooms are the classical examples. Okay. So mushrooms, bracket fungi or puffballs. They will be growing either in soil or on trees, stumps and living plant bodies as well as parasites. Examples are agaricus. All agaricus mushrooms are not edible. Some are very poisonous. Agaricus campestris is edible, but Agaricus sylvaticus is poisonous. Then Astilago, commonly called as smut, Puccinia, also called as rust, are parasitic ones. Okay, so the basidiospores are filling their content when these Astilago and Puccinia are infesting and attacking the plants. That will be causing great damage to the crops. And the last category is deuteromycetes. Deutero means which is left at the end. It is left. So this is including organisms which are not included under these categories, which is leftover. That is why they are called deutero. These deuteromycetes are commonly called fungi imperfecti or imperfect fungus. Why they are called imperfect fungus? Because they have no sexual reproduction. No sexual reproduction is known. If any member is found to be reproducing sexually, then they are all removed from it, because only asexually reproducing fungi are all coming under this category. Here also they are all having branch and septate, hyphae, and they reproduce asexually only. Asexual spores are called conidia, conidiospores. They reproduce only by conidiospores, which are all exogenously produced, and there is no sexual reproduction. That is why they are called imperfect fungi. Now you got a clear idea. What is the name of the group? Common name. What are their important three characteristics? 1. Hyphae. 2. Type of spore formation. 3. Fruiting bodies. Is that clear? These are the three criteria based upon which we are all grouping the fungi, and you will be getting more questions than any other group from this single group, namely fungi. Now, coming to the examples of deuteromycetes, they are the most important saprotrophs helping in the mineral recycling. Examples: Alternaria, it's a parasitic one. Calyptotrichum, again parasitic. Trichoderma, that will be attacking our skin. It is even attacking and causing diseases in human skin. Is that clear? Deuteromycetes. Now we pass on to the kingdom Plantae. Kingdom Plantae include all plants. Don't think that all plants are autotrophs. We are having some parasitic ones, Cascuta, some insectivorous plants which are all eating on insects. Why they are all eating on insects? Because they use insects as a source of nitrogen. They cannot take, they are not getting actually nitrate from soil. They are growing in soil that is very poor in nitrate. So they have to get nitrogen for their need. And for that, they are all trapping the insects. Okay. Generally, they are all having a cell wall and they are all having autotrophic nutrition. An important feature of Plantae is that they show what is called alternation of generation or metagenesis. There are two stages. What are they? Sporophytic generation and gametophytic generation. Sporophytic generation is spore producing, diploid. Gametophytic generation is producing gametes, okay, haploid. These two generations come alternatingly in the life cycle. That is called alternation of generation. Whereas in Animalia, there is no cell wall and they are always heterotrophs. No cell wall, heterotrophs. And these heterotrophs may be either living as parasitic one or they may be holozoic in nature. What is holozoic? Meaning they take complex food material into the body, then they digest and absorb that digested food, and what is not digestible is sent out. Okay. In higher forms, you have elaborate sensory and neuromotor system. They will be having a well-developed locomotor system and nervous system. So they are highly sensitive to the changes in the environment. And sexual reproduction is by copulation of male and female, followed by a definite stages of development called embryological development. Now, in this 5 kingdom classification, Whittaker has not included viruses, viroids, prions and lichens. Why? Because viruses are having no living nature. What is the most important feature? Cellular organization. They are not having any cellular nature. That is why viruses, viroids, etc. are not included. He does not consider them as true living because they don't have a cellular organization and they are all obligate parasites. Why they are to live on the body of other organisms? Because they don't have a metabolic machinery. Because of the absence of metabolic machinery, they will be forced to live as a parasite. And virus means venom or poisonous fluid. It was Ivanovsky who recognized that there are some microorganisms that can cause disease in the tobacco. The disease is mosaic disease. And they were all found to be even smaller than bacteria so that they can pass through bacteria-proof filters. And Bejernik demonstrated that an extract taken from a disease or sick plant of tobacco, if it is given to a healthy plant, will cause it converted into a sick plant. So he called them as contaminated with poisonous living thing. So the term contagium vivum fluidum, the pathogen virus is the reason why the healthy plant is changing into a sick one. And it was Stanley who for the first time isolated them. And when they are all isolated, they are all showing no life patterns. They are all inert and crystal. These viruses are thus obligate parasites. When they are taken out, they show no living properties and they never have any cellular organization. That is why Whittaker is not including them. What is the structure? Viruses are having a core. That core is either DNA or RNA, but never both. And around them, you will be having a protein sheath. That is in brief the structure of a virus. So, viruses are containing either DNA or RNA of the genetic material and they are covered by a protein sheath. That protein sheath may be of different sizes and shape. In tobacco mosaic virus, it is made up of number of units called capsomeres, whereas in a virus that infect bacterium, they are called bacteriophage. It is having hexagonal plate-like structures. They will be all infecting plants, animal or other bacteria and thus they are all living as parasites on them. They will be causing a large number of diseases for both plants and animals. Okay. Now, viroids are isolated in 1971 by Diener. What is the main difference? Viroids will be having no protein coat. They are simply an RNA, naked piece of RNA having no protein coat. And the RNA will be of low molecular weight. They are all living only on plants. They are all plant pathogens, plant parasites living as parasites in plants having only RNA. Prions are abnormally folded proteins. Change in the folding of this protein make them infectious. Such infectious proteins were found to be the causative agent for what is called bovine, meaning related to cattle. Spongiform encephalopathy, commonly called as mad cow disease in cattle, and in man it is causing what is called Jacob's Creutzfeldt-Jakob disease. It is CR Jacob, Creutzfeldt-Jakob. Creutzfeldt-Jakob disease in humans. So, it is similar to the viruses because they can cause other proteins to undergo misfolding. So they are all infectious proteins. That is why it is called prions, proteinaceous infectious particles. Now, lichens are symbiotic association between both fungi and algae. Once you separate them, they become dead. They cannot be separated. They can live only in that association. Once you separate them, they cannot live. So, how can you group them? Can you group them along with fungi? No. Can you group them along with algae? No. So, such an association where both of them are benefited is called symbiosis. And each member is called a symbiont. Algal component is called phycobiont. Similarly, fungal component is called mycobiont. Okay. That fungus is giving nutrients to the algae. Algae is preparing food and giving it to the fungus. So, they are all autotrophic and heterotrophic in nature. Both of them are, see, algae is autotrophs, fungi is heterotrophs. Algae prepare food for fungi and fungi provide shelter and absorb mineral nutrients and water for the partner. So, they are remaining in close association. That if one saw a lichen in nature, that imagine that it is made up of two organisms, but we will never understand that it is so closely associated. One important feature of this lichen is that they are all good pollution indicators, meaning they are highly sensitive to sulfur dioxide. So, in water or air that is polluted with sulfur dioxide, there will be no lichen. Suppose you go to an area where there is plenty of lichens, what does it indicate? That area is non-polluted. Absence of lichens indicate that the area is highly polluted. Is that clear? So, with that, we conclude our present session. I hope it is beneficial to you in every way. Okay, it is in a nutshell. We have prepared the entire content in a small packing. Hope it will be highly beneficial to you. And with these words, I conclude my session and thank you and goodbye until we meet again with the next chapter. Thank you and thank you all once again.